And get started. Hello everyone. I'm Nikki Blunier with the IPM Institute Sustainable Communities Group. Thank you for joining today's webinar, Wyoming's first tick-borne disease surveillance program, presented by Mikenna Smith and Kelsey Mitchell of the Teton County Weed and Pest District. Uh, this project's funded through the North Central IPM Center, supported by the Crop Protection and Pest Management Program project from the U.S. Department of Agriculture's National Institute of Food and Agriculture. And a little bit about the IPM Institute, if you aren't familiar, we were founded in 1998 with the goal to increase adoption of IPM in agriculture and communities using an educational and solutions-based approach. We have two groups within our organization, Sustainable Food Group and Sustainable Communities Group, and the Tick IPM Working Group is part of the Sustainable Communities Group. We also have a program called Pest Defense for Healthy Schools, which is part of the School IPM Working Group, which has free IPM courses for K through 12 staff. And we have Green Shield Certified, which is a certification program for pest management providers and facilities with a mission to reduce the use of pesticides and provide best use practices. And I just wanted to say thank you to our Tick Academy attendees. We had 11 speakers, 114 registrants from 31 states and 8 countries, and 7 hours of educational tick content. It was a big success, so thank you to everyone who made that happen. And I wanted to mention that we are asking for letters of support. The deadline is next week, Tuesday, November 25th. If you want more information about the objectives, abstract, and instructions for writing your letter, you can email me at mblunier@ipminstitute.org. This is for our application for the North Central IPM Center for renewed funding. And with this renewed funding, the working group will continue to host the Tech Academy, quarterly webinars like this one, um, maintain an active website with important resources for integrated tick management, and provide tick collection resources to the North Central communities at risk of tick-borne disease. And the letters of support will greatly strengthen our proposal for renewed funding and increase our chances of being able to continue this work. So again, please email me if you're interested. And if you've already sent one, we really appreciate it. And there's also more information about the objectives and abstract if you are a member of the working group. It's posted on Basecamp. And now I'm going to introduce Mikenna Smith and Kelsey Mitchell from the Teton County Weed and Pest District, and also going to launch a pre-question. We launched these to gauge audience participation and learning. So if you could just answer this pre-question, we'll have the same question at the end, and we'll see like what did you learn?
Oh, that's fun.
So you can go ahead and share your slides whenever you're ready.
Okay, I'm sitting here watching the poll, having a good time. Okay. All right. Can you guys see my slide?
Looks good.
Wonderful. Okay. We'll go ahead and get started. Um, my name's Mikenna Smith. I'll be giving kind of the first half of the webinar, and then I'll hand it over to my colleague, Kelsey Mitchell. And yeah, we're very excited to be here. Thank you for the invitation to speak about our work. We're pretty nerdy and excited about it. Oops, sorry. I'm gonna hide those. Okay, sorry, I had to change my settings real quick. We're very excited to be here. We're very excited to share our work with you guys. Thank you for the invitation. And this is Wyoming's first tick-borne disease surveillance program. And my slides are not advancing. Oh my gosh, we just did this. Did they advance that time?
Yeah, it works.
Oh my gosh, we checked and they even worked and now they're not. All right, guys, we're fine. Anywho, I'm Mikenna. I'm the entomologist here at Teton County Weed and Pest, and I've been here for about 7 years. And my role at the district is vector surveillance, so the mosquito and vector tick surveillance, insecticide resistance monitoring. I run the biological control program for weeds, and I also do laboratory oversight. So that's kind of what I do here at Teton County Weed and Pest. And I'll let Kelsey hop on and introduce herself.
Yeah, I'll echo. Thanks for having us. My name's Kelsey. In terms of the parts of my role that are relevant to this group, I manage the pathogen testing part of this program. So I am essentially managing sample processing as well as new assay development for our vector programs, both mosquitoes and ticks. I also try to find ways to use the lab that we already have for our vector programs and see if we can diversify our capabilities in the lab to also include our invasive species management programs, which Mikenna will speak to in a moment. We'll give you a little bit more of an idea of the organization that we work for in general.
Yeah. So Teton County Weed and Pest, or TCWP, we are a government entity. We're a special district in Wyoming. Every county in Wyoming has a weed and pest district. We exist by state statute, but we're all a little bit different. We each have our own problems and things that we're working with. So for Teton County Weed and Pest, and we're up in the northwest part of Wyoming. Many of you are probably familiar with Grand Teton National Park or Yellowstone National Park. We're near those areas of Wyoming. And this is our mission statement from Teton County Weed and Pest, and that's that we exist to protect, manage, and restore ecosystem integrity through comprehensive invasive species and vector-borne disease management for the benefit of ecological and human health. Um, some of you who are familiar with One Health might think that that sounds pretty similar. I swear we didn't steal it from One Health. We came to it very organically. But I do think it's interesting that independent of each other, we kind of came to a similar goal to One Health. So, and as our name implies, we do both weed and pest work. So on the pest side of things, we mainly work with vectors. Other weed and pest districts do work with agricultural pests, but not so much an issue for us in Teton County. So simply vectors for our pest program, and we do applied research, which really goes between both departments. We have an invasive plants program, which is quite large, and education outreach. So for today, we're going to zoom into vectors, of course. We're going to talk about ticks specifically. And just a note here that of the vector programs that we have, one of them, our mosquito program, has been around for decades, and that's really a mosquito abatement program until more recently. And that's in contrast to our tick program, which is a whopping 3 years old. So it's pretty new, and that's kind of what we're going to talk to you guys about today. The motivation for us to initiate this tick program, no news to anyone on this call, but we're seeing tick-borne diseases increasing in the United States. We're seeing range expansion occurring. But when it comes to Wyoming, it's hard to make claims about these trends because it's very understudied. We don't really know too much about ticks in Wyoming. Before us, there wasn't an entity consistently studying ticks. So we're seeing these trends on a large scale, but Wyoming itself is kind of a black hole of information when it comes to ticks. So that was the major motivation for us to initiate this program. And really on the heels of laboratory capacity building. So we did have a small lab that we were doing some mosquito work, but in order to start playing around with ticks, which carry a higher biosafety hazard than mosquitoes, or they can, we wanted to build up our capacity in our laboratory a little bit. So just a few notes about that. And some background there and where we built capacity. It was both within our facility, our staff, and our programs. So for our facility, our laboratory was, when I first started 7 years ago, was a storage room with a microscope in it. That's the joke that we like to say, because more or less that was the case. But it's been a major goal of mine the past 7 years to grow that up. And so now we're a BSL-2. We have an insectary. We can do a lot more. Um, Laboratory capacity building also meant increasing staffing. For many years, it was just me by myself, and I was absolutely busting at the seams. So in 2023, we hired Kelsey as a full-time biologist to help grow these programs and diversify the laboratory with me, which has been fantastic. We also now have the ability to add on some seasonal technicians, some interns to help us out. And this has allowed us to grow our programs from simply mosquito abatement to include also insecticide resistance testing, RT-qPCR, of course, ticks, which we're going to talk about in just a second, and then also diversify into other stuff that Kelsey's been working on, which has been fantastic, such as genetic ID for invasive plants, but that's a whole nother talk. And this is our laboratory. I hope the slides are, or the pictures are rotating through for you guys. Great. We call it small but mighty. It's not a major space, but as Kelsey likes to say, you can do a lot with a centrifuge and a thermocycler. So think of this as a storage room, and then fast forward, it's our little laboratory now. So this is the kind of space that we get to work in. Okay, so we're motivated to build this program. We've made this lab happen. And so this is kind of where we wanted to start with day one. What were our program questions? What were our research goals? And they were super basic. So again, we didn't have a pretty consistent database about ticks in Wyoming. There were some researchers who've come out and done a study here and there. But for the most part, there's not longitudinal data. There's not a lot of consistent data. So we wanted to start with square one and ask the question, what tick species are present in Wyoming? Is range expansion of non-native ticks occurring in Wyoming? And when I say non-native, I mean not endemic to Wyoming, not necessarily an invasive tick. I hope we never get them though. Also, what pathogens are Wyoming ticks infected with that could be a threat to public health? And how do we communicate that risk to the public? Ticks of medical significance that we are interested in looking for, according to the literature. We kind of have 4 or 5. The Rocky Mountain wood tick, or Dermacentor andersoni, I like to call this our state tick. The brown dog tick, Rhipicephalus sanguineus. We have 1, maybe 2 soft ticks as well, Ornithodoros parkeri, as well as Ornithodoros hermsii. And I have that one in a question mark because there's estimated distribution maps that it is here, but we haven't been able to show that with actual records of establishment using the definition that I'm sure you guys are all familiar with. Establishment meaning more than 6 ticks of a single life stage or more than 1 life stage collected per county within a 12-month period. And then lastly, the American dog tick, Dermacentor variabilis. And you'll notice I have that one also in a question mark because same story as Ornithodoros hermsii. Estimated distribution maps suggest that it is in part of Wyoming, but there haven't been any records to actually show establishment according to that definition. So I'll circle back to that in a minute. Changing lenses a little bit. So same vectors, and these are the pathogens, or excuse me, the diseases that have been implicated or suggested to be transmitted by these species. I won't go through them all. I think you guys on the call are probably pretty familiar. I maybe will mention for the Rocky Mountain wood tick, the one on the far left, it is the only known vector of Colorado tick fever virus. So that's one that we're particularly interested in looking for. Okay, so elements of our tick program are pretty basic. We got basic questions. Surveillance, species ID, pathogen testing, and community education and outreach. So I'll start with surveillance and some of our strategies there. We utilize both active and passive surveillance. I'll go over active first. So active surveillance, the method that we like to utilize is a 30-minute flagging survey that we standardize our effort by time. A lot of you on the call are probably standardizing your effort by unit area using a transect. And for us, unfortunately, that doesn't work too well in our habitat because, as you can see on the top right photo there, this is pretty typical habitat for the Rocky Mountain wood tick. We have this sagebrush slope. A lot of hiking trails are in this area. The topography is very complex. We have these really big woody shrubs, and you can't walk in a straight line, run a transect that way. It just doesn't work. Um, so we have to do flagging, which is more appropriate for topographically complex habitat, but we still wanted to be able to standardize it in some way so we could quantitatively compare sites to each other or longitudinally compare sites to each other over time. So we use a stopwatch with you can see in a little circle there on Kelsey, that helps us quantify ticks per unit time rather than per unit area. So that's the way that we do it. Staff do this active surveillance for us. We also have citizen scientist volunteers who do active surveillance for us. And in 2025, we did a tick blitz. It was our first kind of official tick blitz that we did. And we're not going to talk about it too much in this webinar, but it'll come up in little bits. And so because we utilize volunteers to do active surveillance, we did need a method that was simple and dynamic. And so this 30-minute flagging survey kind of captured that for us. Some considerations with our surveillance strategy that we are aware of but okay with, but just to kind of let you guys know what they are. We do 15 minutes on either side of a trail. So one section of a trail gets done at a time. And this gives us 30 minutes total. And we repeat this on different trail sections. So we just break it out into different, we break a trail down into different sections. Depending on the width of the trail, this is likely an overestimation of encounter risk. If the trail is wide, that's not necessarily the case if the trail is narrow in some case. Certainly, we're going to see variability between surveyors, and our sample sizes between sites are also pretty variable, but we're pretty, we don't want to bias sites that have low abundance of ticks. So we survey sites that have a lot of ticks, and we also survey sites that have low abundance of ticks to avoid biasing against those sites. So some considerations that we're aware of. Our passive surveillance, on the other hand, as you guys know, you don't get as much high resolution data, but you can, it's so easy to still get kind of point presence absence data. Public and partners, they mail us ticks, and it is my favorite thing in the world. It's like opening presents on Christmas. This is me getting some of my tick mail, and it's so fun. People write me letters. I'm such a nerd, but we also see some pretty interesting stuff in the mail. Ways that we've received ticks passively, we've got them off people, by hand, wildlife, pets, and livestock. And again, this only gives us tick species and pathogen presence. You'll see pathogen with an asterisk there because that's only if they meet our testing criteria. We don't test every single tick that comes to us in the mail because a handful of them don't meet our criteria, and Kelsey will go over that in a few minutes. But we still get some pretty good information that way. Real quick about our program, as our name implies, Teton County Weed and Pest, we're based in Teton County, and that was the intention of this program from day one. That was going to be a Teton County program, but it became very popular overnight. Everyone that knew we were doing this, they wanted to send us their ticks, they wanted to get involved. It was wildly popular really quickly. So basically overnight, our program became a statewide program, and we received some grant funding through the Wyoming Mosquito and Vector Management Association to help expand the program to a more statewide level. And so we hope to be able to do that for at least a few years. Okay, so I told you, how we got there and why we're doing it. So let's talk a little bit about some of the data. Again, our program is pretty young. We've only got a few years of data, but we'll share with you what we have so far. Okay, in terms of total ticks collected, no, we are not seeing more ticks every year. There's just this, the program is young. 2023 was really a pilot year, so we were just figuring things out. So we'll probably see this for a while now, the number of ticks that we see increasing per year as our program grows and as people citizen scientists and things like that get involved. But we are collecting more ticks each year as our reach gets larger. So what you can see, though, from this is the majority of ticks that we observe in Wyoming are adults, and that might be in contrast to what some of you guys see in other parts of the country, or you're more interested perhaps in nymphs. But for us, the adults, especially for dermacentors, are more medically relevant to people. They're more likely to bite people than the juvenile stages, the nymphs and the larvae. And for dermacentors as well, they tend to behave a little bit more ridiculously, so we really have a hard time observing them. So we get a handful of them each year, but for the most part, the vast majority of our ticks collected are adults. Breaking that out into ticks collected by method over the past 3 years, no brainer. The majority of them are collected actively because we're actively going out and looking for them. And of our actively collected ticks, the vast majority are flagging. We do have a handful that we collect by dragging. There are a few sites that actually are a little bit more appropriate to drag in Wyoming. And of the passive methods, the majority of ticks we get are off of people, and a smaller amount off of livestock and pets, handful off of wildlife, and then believe it or not, we do get, a couple dozen hand collected. I swear people write me notes like, I found this crawling on my floor. This was in my sock drawer. Like we get a few hand collected ticks every year, which is always bizarre. All right. Switching lenses again, total ticks collected by county. This is super biased towards Teton County. No, there are not more ticks in Teton County, but this is where the majority of the surveillance occurs. So that's an artifact of surveillance, because this is where Kelsey and I are based out of. So, and a lot of our volunteers are there as well. But you can see we do have a few counties with some bigger collections. We have pretty dedicated citizen scientists and collaborators from other districts in Park County, Sublette, Sheridan, Laramie County. So we do have some where we're getting quite a few ticks set our way. Looking at these data in terms of phenology, this tracks with what we would see generally in the literature, but what's a few interesting things here. We're going to talk about misconceptions towards the end of the presentation, but one thing is people, they don't think they need to worry about ticks until like May, which is totally true. The data shows that the ticks are off the charts relative for our area in May, but truly as soon as the snow starts to recede in our area, and we're really high elevation, we're 6,000 plus feet in Teton County. Neighboring counties are 7,000 plus feet. So we're really high elevation. So as soon as the snow starts to recede, say March, the ticks start coming out. So that's important for us to keep track of, be aware of, so we can tailor our public health messaging accordingly. A couple other artifacts here. You'll notice there's a really big peak on May 15th, relatively. That's our tick blitz. So we have 45 volunteers around the state chasing ticks for us. So we caught, over 300 ticks in a single day. So that's what that big peak is. Last thing here, we wear a lot of hats here at Teton County Weed and Pest. As I mentioned in our introduction, we're doing invasive plant work. We're doing all sorts of stuff. We have mosquitoes. So come, mid-July, yes, the tick abundance is dropping off, but our ability to focus on ticks is also shifting. Our priorities have to shift towards West Nile virus and some other things. So our ability to get out and survey come July does become more difficult. So that's something you're also going to see with our data. Okay, zooming into a few select sites that we surveyed in Teton County in 2025. Again, small staff, it's kind of just Kelsey and I doing this. So we had a few sites that were surveyed weekly, one site that was surveyed twice a month, and then a few sites that were surveyed monthly. And you can see them here on this map. And this is Jackson Hole. Some of you might be familiar with kind of a popular tourist area, but that's what this map is showing. It's kind of the Jackson Hole area. This map was originally developed for locals, so they know all of this stuff. So total numbers that we got in 2025 at these different sites. And you can see, yes, we know Henry's Road has a lot of ticks, and Coyote Canyon relative to that doesn't have as many. But if we look at the totals, but as I mentioned earlier, we do want to quantify or be able to characterize these sites by tick abundance per unit time. So we can switch gears and look at that as average density in terms of ticks per minute. So this was a major goal for our program, to find a surveillance strategy that worked for our habitat so we could do this. So we could say, Dog Creek has more ticks than High School Butte. And we can also do this in terms of infectious ticks per minute or per hour. We don't have a slide for that, but I think that you guys generally get the concept that we're going after here. Is we want to be able to quantify risk in terms of simply encountering a tick, and then also be able to quantify encounter risk for potentially infectious ticks. So that was a major goal of our surveillance strategy that we chose. Okay, just a couple more slides on my side, some species IDs, some of the species that we saw. So this is all this, the years just combined together, 2023 to 2025. You'll notice some species have an asterisk or two asterisks on them. Two asterisks is we don't know the travel history. They came off of an animal. We have no idea where that animal came from. And then one asterisk, we knew it was a travel, it was an animal that was related to travel. So Dermacentor andersoni, though, the Rocky Mountain wood tick, this is kind of a no-brainer. This is the tick that we were expecting to see the most of, and we certainly did. And Kelsey's going to talk about this one a little bit more as it relates to Colorado tick fever virus. You'll also notice the American dog tick, Dermacentor variabilis, while not nearly as many as andersoni, we did still see, over 200 of those guys these past 3 years. And if you recall this slide, I had the American dog tick with a question mark because, again, there was never ever, sorry, there had never been record of establishment in terms of meeting this definition for the American dog tick until our data set was able to show that. So we were able to show that the American dog tick, yes, is established in at least 2 counties in Wyoming, Sheridan and Platte County, and then we have a few records of reports, ones and twos and threes from other parts of the state. So those are pretty exciting, low-hanging fruit, but still exciting part of the program to be able to show that with data. Looking at some of the other species that we saw, winter tick, albopictus, that's no brainer. Dermacentor similis, Lone Star tick, western black-legged tick. You'll notice those all have ones, asterisks, so we know those came off of animals that traveled into our state from somewhere else. A few wildlife ticks, a couple of them also, we're not totally sure where the travel history, especially Ixodes cookii. I hope that one was a travel case because I don't want that one here. Ixodes texanus, Ixodes kingi, a spinos ear tick. This one is highlighted because it's the only soft tick we've ever seen, which I thought was really exciting and fun. It wasn't a medically significant one, but it was really still fun to see it. And then another wildlife tick, a rabbit tick. Okay, so that wraps up my portion, and I'll kind of hand it over to Kelsey to dive into pathogens.
Thanks, Mikenna. Yeah, so we wanted to include in our program, in addition to surveillance and species ID, pathogen testing. And we felt like we would be able to do this because we already had enabled our lab to have capacity for qPCR-based testing of mosquito pathogens. And with a couple of safety upgrades, we were also able to do that with ticks. So next slide, please. Perfect. Thanks. Good job. So First, we're bringing back this slide so that we can look at, okay, what are the medically significant ticks that we now know are here, and what are the illnesses that they can vector? Those are listed below each species. So there's a lot of things that we could dig into, a lot of illnesses caused by different pathogens. And it's just McKenna and I, and really it's just me doing the pathogen testing. So we really needed to prioritize where we wanted to start in the first couple of years of our program. And we chose Colorado tick fever. Reason for that being one, as you saw from our surveillance data, the number one tick that we found, or tick that we found in most abundance, was the Rocky Mountain wood tick, which is the only known vector for CTFV. Also, while there is quite limited or sporadic literature for tick-borne pathogens in Wyoming, the literature that is available suggests that Colorado tick fever virus is a good place to start because Wyoming, particularly Sublette County, which is the county in Wyoming with a little star in it in this map, has the highest human case rates of Colorado tick fever in the United States. And that is despite there being areas, other states that actually have higher infection rates among ticks. Sublette County still often has the highest human case rates. And something I think that's interesting, and I wonder if this is different from kind of the attitudes and practices that you might encounter in the East. You'll talk to people, right? We'll talk to residents of Sublette County. McKenna is a resident of Sublette County, and you chat with them. And they just expect to get sick if they get bitten by a tick. It's an expectation. Oh, I'll just get tick fever. So they just call it like a general tick fever. So it's likely vastly underreported in Wyoming. Yeah, it's an expectation. I'll just get sick. Colorado tick fever virus specifically presents as an acute febrile illness. It often will present in a biphasic nature. So you get sick, feel better, get sick again. And for most people, it's just going to be like a bad flu, but there's an estimate of about 15 to 30% of cases that do result in, or reported cases at least, that do result in hospitalization due to encephalitis, and that's going to be in the older and the younger members of the population. All right, a little bit more background because I think Colorado tick fever virus is maybe a little bit of an obscure pathogen for folks in this audience, maybe not. There's a couple people I see in the audience who know way more about it than I do, so bear with me. No, CTFV is a member of the cultivirus. It's as the name shows, it's the prototypical virus in this group. It's non-enveloped, and the genetic material is a double-stranded RNA. Something I think that is also important or unique about this pathogen in terms of the full system, the ecology of the system, is that it has numerous mammalian hosts across several different genera. And so there's a few different species of ground squirrel, chipmunk, mice, wood rats, porcupine. There are quite a few different hosts. And so overwintering does occur because the hosts maintain persistent viremia for weeks to months, and there is transstadial transmission across the Dermacentor andersoni life cycle. All right, how do we detect this pathogen in our ticks? Again, for most of you, this is going to be a review, but we use reverse transcription qPCR. Our workflow is shown here on this slide. We take a tick, we actually cut it in half. We reserve half for later for long-term storage, just in case we not just in case, for when we definitely want to go back and look at those ticks for potentially other pathogens or other projects that we just haven't been able to dream up quite yet in the first couple of years of this program. So we reserve half the tick. The other half is exposed to a dual inactivation with both heat and chiotropic salt. It's homogenized. The viral RNA is extracted, and then that double-stranded RNA is denatured immediately prior to RT-qPCR. And not every tick, McKenna talked a little bit about this, not every tick is tested. They do have to meet our testing criteria, which are here on the slide as well. They need to be Dermacentor andersoni. Eventually, who knows, maybe if we have some time that we find at some point, we'll test some others for fun. But right now that doesn't make sense because Rocky Mountain wood tick's the only known vector for CTFV at this point. Um, we don't test blood-fed ticks because partially because of safety and exposure risks, but also because blood is known to inhibit PCR. We only will test ticks that have arrived to our lab alive. So that's that pathogen. No, sorry, that's the passive surveillance side of things. We're getting ticks in the mail. Are they arrived to us crispy and dried and gross and dead, or are they alive? So to ensure that we have quality RNA going into the workflow, we only test ticks that have arrived at our lab live. And those are either tested immediately or stored at -80 until processing. Um, so of the 3,100 about Rocky Mountain wood ticks that we have found or collected rather in the first 3 years of the program, 88% of those, so just over 2,800, met our testing criteria, which I think is pretty exciting that many ticks are hardy. They can really put up with a lot. And so them coming through the mail, we were quite pleased that many ticks met our testing criteria. To date, 1,225 ticks have been tested. And then I'm going to talk about the data, and I'm going to talk about it in two different kind of chunks, 2023 and 24 ticks together, and then 2025 samples. The reason for that is because we had processing changes like on intentionally from 2024 to 2025. So in 23 and 4, we had about 750 individual ticks that required testing. And so I was able to do those individually, one by one. We also wanted to see that's a number that's doable by hand, one by one. But we also, for those first couple of years, wanted to have the finest resolution of data that we could because it had been 10 years since anybody had looked at this virus in ticks collected from Wyoming. In 2025, we did switch to pooling ticks up to 5 because we have about 2,500 ticks to test from just 2025. And I'm pretty good, but there's no way that I can get through that many ticks individually by myself. So we will unpool any positive pools and still be able to get that individual infection rate. So for 23 and 24 ticks, 48 ticks of the 747 tested were positive. And you can see here the map that would communicate that data out at the county level. So kind of higher resolution, but as you all on this call are very familiar, this is typically how arboviral data is communicated out and tick pathogen data is communicated out at higher levels into public health. So we have some interesting information. We have some varying, it looks like some really drastically varying infection rates of Colorado tick fever virus. But when we're most interested in risk and communicating risk out to the public, and so looking at the county level doesn't really give you, I don't think, a full story when you're interested in risk. So zooming in here for our 23-24 samples to Teton and Sublette counties. And actually, these are just portions of Teton and Sublette counties. And if you'll remember from the previous map, Teton, oh, thanks, Mikenna. Teton and Sublette are neighboring counties, with Teton County being, Grand Teton National Park is a part of it. Yellowstone is just north of us, and then Sublette County is the county that's just to the southeast of Teton. So when we zoom in, you can see each little pie chart here is a site where ticks have been collected from. Blue is Colorado tick fever virus negative, yellow represents positive. And so, yeah, the Teton County infection rate county-wide is 0.5%. But if you look at each site, you'll notice in these data, only one site was positive, but that site had a 9.5% infection rate. Sublette County is a little a little bit of a different story, but higher countywide rates, 17.5%. But we have these sites that are that have really high infection rates or relatively high infection rates. Of course, you'll notice the two sites at the bottom that have positive ticks are have an extremely low sample size. But I think that what's really interesting is when you look at those, and you think about how many ticks you would typically have to sample to find X infection rate. We're getting ticks from Sublette County that somebody happened to find on themselves hiking with their dog. It's a one-off, and that one tick happened to be positive. So we're really interested in spending more time in these counties, spending more time in these areas, and trying to have a better resolution of the data so that we can understand more about the nature of this system. Is Colorado tick fever virus very, very focal? Is it, do we have these hotspots? Do we not? We don't have enough data to answer any of those questions yet, and we'd like to. So we're going to keep drilling into that. But I just wanted to show a general example of kind of that county rate versus site-specific rate. Moving on to 2025, I have a lot of testing still to do. So I've tested 113 pools comprised of 478 individuals so far. 13 of those pools were positive across 4 sites in 3 different counties. And so you'll see this map here again, showing that county rate. A couple things that I want to note here. If I were in the audience looking at this, I'd be like, wow, from 23 and 24, you had a 0.5% infection rate in Teton County, and now all of a sudden, with really not all that many more ticks, about the same number of ticks tested, you have 10%. What's going on there? No, it's not just like, wow, all of a sudden we have all of this virus. I focused on the sites that we knew to be positive first. Or not new to be positive. I focused on the sites where we previously had known positive ticks first. And so this is likely very, very overestimated. But I wanted to focus on those known positive areas first so that we could communicate that out to our partners in public health and start to think about what our messaging should be to the public. So that's likely to decrease over time as I do lots and lots more testing. Again, this is more for folks who are familiar with the area, but I also focused on testing the weekly sites, our weekly surveillance sites first. And so you'll see we have two sites that we know to have positive ticks. Those are High School Butte and Coyote Canyon. If you're ever in the area and want to avoid them, don't go there. Just kidding, still go there, but protect yourself. So we do have a couple of ongoing collaborations that we're pretty excited about. In addition to needing to do a lot more testing for our 2025 ticks. Couple of collaborations which have been really amazing because we don't have the capability to do either of these things in our lab. We partnered with the CDC to do viral isolation from 15 of our Colorado tick fever virus ticks. They have gotten through 10 of them at this point. And this is work that Joni Kenny is doing. They've gotten through 10 of those 15 ticks at this point, and 8 of them they've been able to successfully isolate virus, and she's pretty excited about how that project is going so far. We're also collaborating with Wyoming Public Health Lab and they have been helping us with a couple of different things, but one of them, which is pretty awesome, and we would not have the capacity to do this in our lab, is an Illumina viral surveillance panel, which screens for over 200 pathogens. And so some of that was a little bit of proof of concept. Okay, we have these known Colorado tick fever virus positive sites. The panel should pick that up. Does it? The answer is yes. But also we have all of these ticks that are Colorado tick fever negative, but I've only tested for one pathogen. And we have a long list of pathogens that we would like to ultimately test for. But how do we prioritize those? This panel could really give us the power to have a data-driven approach that is from regional ticks rather than just looking at the literature and guessing what's the best to work on next. This panel can give us a data-driven way to start to prioritize our assay development for subsequent pathogens. All right. I feel like I talked really fast. Sorry, guys. Our final part of the program, and really the whole goal of this program, right? That's the whole goal of knowing what's here, knowing what it carries, is to be able to communicate that risk out to our community, which, we have both residents and we also have millions of visitors that come through this area every year. So, this is definitely, I would say, our community education and outreach at this point in the program is the least formally developed at this time in terms of our program. But that makes sense. We had to start somewhere and we had to get information to be able to communicate out. So our first couple years are getting that information, but we're really interested in growing this part of the project and the program. And so, really, with our overarching question being, what are the attitudes, knowledges, and practices of the community regarding ticks and tick-borne diseases? And once we know that information, how can we affect meaningful behavioral change that is going to keep people safe? Both, again, people who live here and people who are visiting. And so, yeah, identifying best communication practices for that. And then, of course, to continue to spread the word and engage more participation in the tick program so we can get ticks from more parts of the state. There's 4 counties that we don't have any ticks from, and we'd really like to fill in our Wyoming map and increase participation. And we also would like to identify and then address misconceptions. And something that I didn't anticipate as part of this program, or at least as part of the field work of this program, is that we get a really unique opportunity to connect with individuals on a one-on-one level. And of course, yes, the ultimate goal in this program is going to be communicating out at a way higher density than a one-on-one level, but we have been able to chat with quite a few people as we've been doing this work, right? You look kind of weird. You're walking around with a white piece of flannel, rubbing it on the bushes as you're walking through a trail. And the guy who's mountain biking or walking with his dogs, or the gal who's riding her horse asks, "What are you doing? This is odd." And so we get a lot of opportunities to talk with folks one-on-one. And one of the things that has allowed us to do is to identify some of the misconceptions that folks have. And that's listed here in this table. So we'll just go through this briefly. We get a lot of folks that are like, "There's no ticks in Wyoming." We're like, yes, there are. A lot of times I feel like those people are folks that have moved. I'd love to quantify this, but I feel like those are folks that have moved from other areas, like maybe in the Northeast, where tick messaging is so much more robust, and the rate at which you encounter ticks might be higher. And so, yeah, there's no ticks in Wyoming, and it's super, super fun to pull out your vial of live crawling ticks that you just collected, because, yeah, you watch somebody's mind change right in front of your face, and that's a pretty unique thing to be able to do. We also have because there's no Lyme here, I can't get Lyme here, there's no danger. Obviously, that's not true. There's other things that can make you sick and quite sick. I've been told by my vet I don't need to put my dogs or pets on tick prevention meds. Well, might want to consider it. We've also Mikenna has done a really good job of reaching out to vet offices to both get those travel case samples so we can understand what ticks are coming into the state on pets and on livestock. And so we're interested in building that partnership with veterinarians to be able to make sure that everyone is aware of the risks that we are seeing, the ticks that we are seeing. Oh, when I find the tick, I always just burn it or crush it between my fingernails or slice it with my knife, and then I make my peanut butter and jelly. Like, okay, cowboy, just maybe don't do that. Don't do that, firstly. And secondly, please just bring us your tick that you find. So yeah, misconceptions about the best way or the most safe way to remove a tick or, deal with a tick when you find it. I want to send my embedded tick immediately out for testing. Can you guys do that? This has been I wouldn't say necessarily a challenge of our program, but something that we are definitely taking into consideration is that we can't test blood-fed ticks. So we get a lot of folks that want to send us their tick, but then are also like, well, you're going to test it for me, right? So being really clear about communicating what services at this point we can and cannot provide for members of our public has been something that we are working on as part of communication. Mikenna touched on this one. Oh, ticks aren't out till May or June. I don't need to worry about them until then. Our data suggests otherwise. Oh, you can get Lyme in Wyoming. We hear that one sometimes. there's no entomological evidence that Lyme exists in Wyoming, right? So that's always an interesting one. And then, yeah, we also get a lot of like, well, my aunt who's never left the valley was diagnosed with Lyme. And we find that one, we're the messaging that folks in the Northeast are doing fabulous. Everyone is aware of Lyme. Everyone knows about it, which is fantastic. But it's really interesting out here how that plays into communication and outreach. We tend to typically just ask those folks more questions. Hey, do you mind if I ask you a few questions about that experience? And so we find that, shutting down that conversation with like, oh no, you didn't, there's no Lyme here, is not helpful. And so asking more questions and tugging out about like, okay, this person is really sick, what's going on, has led to some really interesting conversations and some interesting hypotheses about what could be going on. So this is just kind of a subset of the misconceptions we've heard, and that's where and those that we need to correct, and the best ways to do that are kind of our primary next steps. So with that, in a effort to stay on time here, I will pause. And I know I saw a couple questions I think already in the chat, but we would love to take your questions. Thanks so much for your attention.
Thank you so much, Kelsey and McKenna. Awesome job. Great presentation. Really engaging. And I learned some new things.
Right?
Um, so I'm gonna actually share the post question um, and see how people answer this time. But you can we have 10 minutes. You can answer some of these questions.
Great. McKenna, do you want to take how often do you stop and remove attached ticks from the flags?
Yeah. We do it about every 10 to 15 paces. Um, Maybe closer to the 10 to 12 side. The shrubbery, the woody vegetation that we have is it's woody vegetation. It's very rough. So if you go too long, you'll be flinging ticks off of your flag. So about every 10 paces. Is what we do. Mel, yes, the Dermacentor variabilis for Colorado is pretty new, and it was interesting to see how there's no Dermacentor variabilis in Wyoming, in Colorado, and then as soon as you do some surveillance, it's like, just kidding, all of Colorado has variabilis. So, this is the importance of surveillance and why we're doing this. Let's see. How did you ID the albopictus and what life stage were they? I used a key just on the microscope. The albopictus are morphologically, I think, quite distinct from the other dermacenters. Their sporacular plates are, especially the goblet cells on their sporacular plates, are massive, and we don't really have any other dermacenter in our area that look quite like that. So, in contrast to Andersoni and variabilis, they're really distinct. And the key that I use is the one published by Brigham Young University. I can send it to anyone who would like. Um, and what
And then
else?
Mel has a question about hosts. Are there more ground squirrel species near the positive sites versus negative sites for CTFV? Oh boy, we have so many questions. We have so many questions about hosts and the impact of hosts on, yeah, those sites that we see that are you know, for either very positive or very negative, which is a silly way of putting it, a very anecdotal way of putting it. But I can tell you, some of those sites that I've tested from that are positive, I've tested, 200, 300 ticks from a site, and I've never seen a positive tick from that site yet. Meanwhile, yeah, there's another site I've tested, 40 ticks and there's a 20% infection rate. So it's really fascinating. And yes, we would love to look at the answer to your question is we don't know, and we would love to look into that. We suspect that yes, it is. Those sites might be the reason where there's hotspots is going to be closely associated with the hosts that are there, but we haven't been able to do that data yet. And even like anecdotally, I can't really speak to that. I don't see a lot of. I see a lot of burrows, but I don't see a lot of the mammals themselves when we're out collecting. McKenna, would you like to add anything?
Yeah, one of the sites in Sublette County, the one that was a what 27% infection rate with N of 228 or something. The gentleman who owns that property actually shoots his ground squirrels, but he thinks the cottontail rabbits are so cute he won't shoot them. And I'm just like, dude, those rabbits have CTFV, and this gentleman has gotten the disease. So, and we know that CTFV is there's so many reservoir hosts. So, um, and some are better at amplifying than others. So I think it would not be unusual for us to see that one site has a high level of cottontail rabbits that are supporting viral replication. Another site is ground squirrels, so that's an element that I think we're going to be investigating earlier is um, trying to quantify that and trying to collect some host data
We,
without going too crazy.
I know, yeah.
Or maybe we will. Maybe we just will.
Let's go crazy. It's just the two of us. Let's go crazy. Something else that I think is interesting about these sites is that I think they differ quite a bit in the level of disturbance that they've experienced. But again, like that's not something that we've looked into in particular. But a few of the sites I would say are pretty environmentally disturbed sites. Quite a few invasive species, highly trafficked. Um, and some of the other sites are quite a bit less disturbed, more intact, native vegetation, less off-trail use, things like that. So we're also curious about what impacts that those indirectly have. Mikenna, Laura wants to know if when you were looking under the microscope, she said apparently the rabbit tick has been confused with Asian longhorn tick in the past. So when you saw it, did your heart jump at first, or are you good at saying immediately it was a rabbit tick?
No, I was not good at immediately saying it was a rabbit tick. I could immediately tell it was Haemaphysalis, and I was like, "Oh no!" I knew we had wildlife Haemaphysalis. I absolutely knew that, and so I was like, "Settle down. You're fine. Everything's fine. Break out the key and sit at the microscope for 5 hours to be sure." And then, sure enough, it was Leporus palustris. But it was cool to see the Haemaphysalis, and I did have a reference collection of uh, Haemaphysalis longicornis to confirm against. So, yeah, and that one we had Kelsey had actually caught that one. We were surveying the gentleman's property who has a 27% infection rate, who have all these rabbits that just like live in the peri-domestic environment. And so we were kind of trying to see if we could get some immatures because these rabbits were using like his wood pile and his like scrap metal pile. And it was kind of a little bit of a decrepit, um, storage environment. And so we were trying to see if we could find juvenile Andersoni associated with these rabbits using these structures. And then so I was also a little bit less scared knowing that rabbits were at the site where that Lepora palustris was collected. So that gave it away a little bit that the hosts were right there. Um, let's see. That would be great. Part of your vector surveillance, Krishna is looking for albi. Oh yeah. Finding the engorged albopictus on moose bed sites. We got so many this year. Oddly enough, just like everyone was like, the moose is in my yard and it dropped off 20 ticks. Do you want them? I was like, you betcha. Um, so that's same thing. That's about the only time we can find them is when they're dropping off the moose in the spring. And I have quite a few in collection if anyone would want any. I have quite a few Andersoni as well, especially the ones that didn't meet testing criteria. I know all you nerds probably have your tick collections, but if you're outside of the West, you probably don't get to see Andersoni. So if anyone wants Andersoni for their collection, I've got all life stages. So if you want to email me, I'll send you some. Or Albopictus as well, but that one's not restricted to the West.
Thanks.
Zach,
Zach,
you
you
raised your
can
hand. Oh, go ahead.
ask your question.
Just really quickly, for my own passive surveillance program, we have our residents drop off their ticks, um, just out of concern of what ticks desiccating not being able to be identified or being crushed in the mail. Is that something that you guys have run into pretty often with your program?
Them being desiccated and not identifiable.
Yeah.
Oddly, no. Like maybe one tick I've ever received that was glued to a microscope slide. And I was like, scientifically great effort. Love that for me, but I can't see anything under this layer of glue. So there is only one tick I've ever been able to not identify. So, but our ticks, I don't know, they're mostly adults, so they're big, and the dermacentors are quite big and they're pretty robust. We have people, what we ask people to do is like get a piece of tape and put it on, either like fold it inside of itself because they will desiccate. And that's really what we're most interested in is getting them alive. And it seems that the loss of humidity is what kills them in the male. So putting them inside a piece of tape and like holding them in their own little humidity bubble, or putting them in a Ziploc bag with a few blades of grass seems to work really nicely. If they're totally engorged, they'll pop in the mail if they don't get put in like something. So a few of them come to me, like they've been through a roller, but if I can see this brachialar plate, I can still ID them. But um, yeah, it's so far actually it's been surprisingly successful in terms of at least getting to get species ID out. So I hope that
Right.
helps.
Hey, that's
It
Yeah.
good.
does. And I'd love to get some Andersoni, and if you want, I can send you a ton of Asian Longhorn tick because I've
Oh,
seen just so many of them.
yes, that would be great. I would love that. I'd be happy to send you some. I've got a bazillion.
Got another comment in the chat.
Oh, that's wonderful.
Oh, thanks, Mel. You're a fantastic audience member. I could see you nodding along. I was like, she's with me. I got it.
Oh, that's fantastic. One of these days, I've been telling Kelsey, we didn't talk too much about it in this webinar, but we did a tick blitz and maybe in a couple years, it's a big pipe dream to do a multi-state tick blitz. And I've got contacts in Colorado, so I'm thinking like a Colorado, Montana, Utah tick blitz. So you can um, you'd be hearing
Yeah.
from me if I ever get to pull that off.
Awesome. Well, it is a couple minutes past 2, so we're gonna wrap up. And both of their emails are here in the chat. If anyone has more questions or if you want to send them to me, I'll forward the questions. Um, Thanks again, Kelsey and Mikenna. Um, Thank you everyone for joining us today and see you at our December webinar, which I'll be sending out more information for soon.
Thanks for inviting us.
Thanks, y'all.
Thanks.
Bye.
Bye.